We're going to talk about the gut microbiome, its composition, and how that composition is an indicator of early Alzheimer's disease. And we're utilizing one particular study, of which, by the way, there are many, but this particular study is quite recent, just came out a couple of months ago, and it's done by a team in the US. And what they really came up with as a fundamental part of this study is that the change in the gut microbiome composition correlates well with beta amyloid and tau pathological biomarkers. And most of you who've read about Alzheimer's know that it is those plaques, the beta amyloid and tau plaques, that are the causes, we think, of Alzheimer's disease. And so we know that the gut microbiome correlates well with those biomarkers, but not with biomarkers of neurodegeneration. Now, that's important because it suggests that the gut microbiome may change early in the disease process before clear neurodegeneration has taken place. That's very important, and we'll go over that over the next few minutes. So if you look at the progression of Alzheimer's disease, we're going to call it AD, the stages of the pathobiology. You begin with someone who's cognitively normal, no evidence of disease, but we know from some of the biomarkers and imaging that the disease has actually started, but it's in its early stages. You then go on to the next step where there's an apparently cognitively normal, but now with classic biomarker evidence of disease. They've entered from that very early stage into a classic preclinical AD. And then, unfortunately, if you wait long enough, you end up with enough neurodegeneration that you're into symptomatic AD. So the transitions from one stage to the next is based on the presence of these markers in terms of us being able to see those stages. And it's identified by a whole number of imaging techniques, PET and other images, as well as cerebral spinal fluid, CSF, these assays. And what are you trying to do? You're trying to detect the pathogenic protein plaques, the beta amyloid and tau proteins, and also markers of neurodegeneration, which you're identifying as well with various imaging. Now, we do know that the gut microbiome, there have been other studies over the years that have shown that the gut microbiome may play a role in early AD. And that composition of the microbiome of people in the earliest stages of AD, when the brain changes have already started, but before cognitive symptoms have become apparent, that this differs from that of healthy people. So, oh, okay, maybe the microbiome is different. Let's take a look. So we know that there are specific gut microbiome types that are associated with preclinical AD. And analyzing an individual's gut microbiota may identify them. Are there greater risks for them of developing dementia? And can it help us to design a microbiome altering system to prevent treatment that's going to avoid cognitive decline? Because that ultimately would be the wonderful thing. Can we, if we know that the microbiome is getting altered, can we devise preventive treatments of the microbiome to avoid that cognitive decline? So there's a gut-brain interaction here, but we do need to ask, is the gut influencing the brain, or is the brain influencing the gut? So these authors said changes in gut microbiome may be just a readout of pathological changes in the brain. Maybe it's the other way around. Or the alternative is really that the gut microbiome is contributing to AD, in which case altering that gut microbiome with probiotics, with fecal transfers, might help to change the course of the disease. So that's really the question they're trying to answer. And one of the important things to know is, how long does it take to develop AD? So if you're looking at pre-symptomatic phase of AD, the early stages of it can last 2 decades or more. So those clumps of amyloid and tau start accumulating in the brain, but affected individuals aren't yet exhibiting signs of neurodegeneration or of cognitive decline. In fact, the data suggests that it takes at least 10 years between the first plaque deposition in the brain and to see those first clinical signs of impairment. 10 years or greater. So that gives us a wonderful window of time to be able to change the pathology and to change the actual aspects of AD. So early detection of molecular hallmarks of AD pathology remains critical for implementing any number of effective treatments. And if you want to know, is it really true that bacterial species are associated with AD? Well, if you take a look at this study, it's very elegant. They show all kinds of different ways of determining which one of the various taxa and groupings of bacteria are showing that. And here is one of the things they show in that study, that markedly different gut bacteria in terms of bacterial species and the biological processes of these bacteria are showing in preclinical AD as opposed to healthy. It's very clear. And that is clear even if you change diets, and if you have the same kind of diets, doesn't matter. They show up. So the microbiome differences in healthy and AD individuals is real. The difference in the microbiome composition and microbial functioning at these preclinical stages of AD could therefore be used to screen for early AD. Minimum, screen for it. Even if you're not going to be able to treat it with changes in the microbiome, you can at least screen for it in a very non-invasive, very cost-effective way. So the association, these authors say, of gut features with definitive molecular hallmarks of early AD pathology strengthens their potential utility as a complementary early predictive biomarkers. And that's very important because you've got to get something that can be done easily, quickly, non-invasively, and very cost-effectively. So if you look at the future, if there is a causative link, most likely that link would be inflammatory. Big surprise. Inflammation raises its ugly head again. And the advantage of gut microbiome, as I said, as a screening tool, is its simplicity and its ease. You provide a stool sample, you find out if you have increased risk for developing AD. It's much easier, it's less invasive, more accessible for large populations compared to brain scans, spinal taps. These are very invasive and they cost a fortune. So you could have treatments as well that promote good bacteria or getting rid of the bad bacteria. And therefore you slow or even stop the development of symptomatic Alzheimer's disease. So what are the key takeaways? Healthy individuals and those with preclinical Alzheimer's disease have harbored markedly different gut bacteria. And the findings suggest that it may be feasible to analyze an individual's gut microbiota to identify people at higher risk of developing dementia. And by doing that, you can design, very possibly, microbiome altering preventive treatments